Thank you first for the invitation and the opportunity, Bill, and I think as Antoniana for the opportunity to present and some part of our research in our lab. So we're going to talk today about GenEPI. So this is based on Piezo1. It's a fluorescent report of visualizing mechanical stimuli. And briefly, everybody calls me Lucky. So it's from Periklis and short Periklis Lucky, also small Periklis, and the short version is Lucky. So in general, then people call me Lucky. So I'm a reader in advanced optical precision imaging at the Department of Bioengineering at Imperial College London. And what we do in our lab is to conceive and apply advanced imaging technologies for a mechanistic understanding of development, disease progression, but also use them also for diagnostic purposes and as well as for therapy, so for more medical outlook. So we have three pillars in our research. One of them is prime conversion, which is going to show only very, very briefly. The second one is GenEPI, what I'm going to go into much detail for visualizing then mechanical stimuli. And finally, which I will not cover, but I would just put like 2 sentences on them, is bioharmonophores. These are biodegradable imaging probes, which can detect single cancer cells in vivo. And the idea is to use them for diagnostic purpose, but also then for therapeutic purposes, so photodynamic therapy or even potentially photoimmunotherapy in the future. So talk about GenEPI. First is what motivates us in some ways is to interrogate biological systems. And in this case is obviously based on GenEPI. We talk not only about chemical interrogations, but also or chemical information, but mechanical information. And if we talk about interrogation of biological systems, one system I've worked on once is the mouse embryo development. So it's how does a single cell develop into a complex multicellular organism. So we previously, a few years ago, supervised work where we looked at transcription factor dynamics and how they actually can predict a lineage allocation very early on. This was done with Nicolas Placht, and he has also now continued to do much different work as well. So here now, the question is, if you look at this mouse embryo development, it starts from fertilization, 2-cell stage, 4-cell stage, 8-cell stage, so cell divisions, and later on you have this compaction, the rounding up at the molar stage, and then the blastocyst. Are there mechanical cues? Do they play any role also during during development, and obviously also asymmetry generation? So previously we have shown transcription factor differences. Other people have done epigenetic changes. Now, recently, there's a paper also from Nicolas Plachter talking about cytoskeleton dynamics as well, which also predispose cells then to different lineages. So the inner cell mass, which gives rise to the embryo, or the trophectoderm, which gives rise to the placenta. So mechanical cues is the focus, and we wondered how to visualize this. There has been already indications by Jean-Pierre Maitre, I guess his name, showing that there's indeed also surface tensions at the 8-cell stage, which also predispose cells then later on where to allocate then at the molar stage, to be outside or inside. And there's been also implications, correlations, to be precise, on hippopathway and YAP translocations, either outside the nucleus or inside the nucleus. So we wanted to see, is there already mechanical changes much earlier? And so this was the motivation, how to visualize mechanical forces. And if you want to visualize them at some point, we would like us to see all the cells, how do they end up? So really a holistic or a comprehensive systems approach of appreciating development. So the fidelity, the high fidelity lineage tracing in some ways, tracking all the cells during development, and let's say the evolution of mechanical cues. Well, as I mentioned, these questions drove us, and we developed actually tools for this. And we hope now to go back and to use these tools now to comprehensively understand this biological system. So, but in general, interrogating biological systems you can do with 2 cases. In one case, it's the chemical approach, and there's also the mechanical. And we know that there's now also interactions between both of them. And in mechanical tools, what we have done is for lineage tracing allocation, is to address one challenge. As you can see here, on the right side is a mouse embryo, the first 3 days of development with a light sheet. It's being labeled the nuclei with H2B. And as you can see, it shows confounded rotation spatial drift. This is not rare. So this comes, 45% of the embryos do have this, show this. And this potentially might be also a mechanical one, right? Cytoplasmic streaming, some changes which might drive them, which is so far not understood really. But of course, it poses a lot of challenges to be able really to follow each and every cell, not just a few of them, to really have a systems understanding. So what we did then is to introduce a methodology which we called primed conversion. So this is a way of photoconversion, so changing a green fluorescent protein to a red fluorescent protein. It is being known originally that you can do this with near UV light. So as you can see here, this is the black arrow with 400 nanometer light. So near UV or ultraviolet light. What we introduced is primed conversion. So the combination of blue light and red light results in the same outcome then. So you have then a red fluorescent protein. The beauty of primed conversion is that you can make an a cross-section setup, so a cross-beam setup at the focal plane. And this then allows you to primed convert, as we now term in the photoconversion, very precisely in three-dimensional space, and also not with the 2-photon laser, which doesn't work anyway, but with low energy, so with continuous wave, and very precise, non-toxic primed conversion. Here you see it in the lower panel is the mouse embryo, 4-cell stage, and one of the nuclei have been primed converted. This is the histone labeled with a primed convertible MESFP. So these are proteins we engineered which are much brighter, much more photostable. With this now, what we then accomplished is with an algorithm, we call them primed track, is to correct for these spatial and rotational drifts of the mouse embryo, which potentially also for organoids. I think they even spin much, much faster. On the upper side, you see, upper panel, you see the movie over 3 days where we prime converted one nuclei at the 4-cell stage. These proteins are optimized. So these are this prime convertible MESFP. So they're much brighter, they're more photostable. So the contrast is higher in the red channel. And so we can track them with only one prime conversion up to the blastocyst stage. But you can see that there is a lot of rotation and drift. And the lower one, what it is to use then this color to use it as a fiducial marker to correct them for these rotations and movements. And as you can see, it's pretty static. And it's very straightforward now to track each and every cell, and also is dynamic if you would like to combine, let's say, with transcription rate dynamics or with cytoskeleton reorganizations, or maybe visualizing mechanical cues at particular time points. Prime tracker allows you to have a high fidelity lineage tracing with limited scans. So usually light sheet, you do it 5 minutes or every 7 minutes. What we did here is 20 or 40 minutes, I have to check again. And so this is a big spacing, but still we have high fidelity lineage tracing. And first, you don't have so much data to handle, and you still have your high fidelity lineage tracing accomplished. Now, this is just only to have a comprehensive understanding of development, to follow each and every cell. Now, coming back now for our methodologies, we would like to interrogate these biological systems from the mechanical point of view. And this is accomplished with different methodologies currently, which I've listed on the left side. You see in one case, you have atomic force microscopy. Then we have droplet-based sensors. So this could be liquids with iron liquids, which actually deform, and this also inform you about the mechanical forces surrounding them. Optical magnetic tweezers. And finally, genetically encoded are the thread-based tension sensors. These ones are very precise, so they have a though very small dynamic range, but very precise. The limitation of them is obviously indeed is that they are very restricted to their particular applications, so these cytoskeletal structures. So now comes into play then Sina, so former PhD student in my lab, who thought she would like to visualize mechanical forces, but not as restricted as these thread-based tension sensors, with high dynamic range and very flexible to be applied in different tissue structures. Now, she then suggested to look at Piezo. Piezos are stretch-activated ion channels, which have been the first time characterized by the lab of Adem Pataputian in San Diego. There's been an explosion of research on Piezo. This is just a review which has been probably used 2 years ago. They're much, much more than functions. But physiologically, you can see it in a lot of different environments. So it is involved in pressure sensing on the left upper side. On the lower ones, it's the bones. So it's for also compressions for bone structures. And then it's important, let's say, in a stem cell environment, on the upper right side, you see it's differentiation of neural progenitor cells into astrocytes or neurons. Another interesting observation is that Piezo1 in particular is massively accumulated in red blood cells, and it's also implicated in the osmotic pressure regulation. It's also in the lower panel, what you see then is also expressed in kidneys, and there it also is based on shear stress, also senses then radial pressures as well as osmotic pressure. Now it has a lot of function and also has been implicated in its function in differentiation, a lot of different pathways in different cell sections. One here is neuronal differentiation. So obviously the point is, what is Piezo1 and how to visualize then the mechanical forces then applied and being sensed by Piezo. What you see here is now the structure of Piezo1. On the left side, you see the top view of this transmembrane protein. So it's top view within the membrane. You can envision this one. On the right side, you have the cross-sectional view of Piezo. On the left side, you see it's built of 3 Piezo proteins, so it's a triskelion. And the right side, you see it has a dome-like structure. So when a mechanical force are being applied, then it actually it's being elevated and levels out, and this then leads into the opening in the center of these triskelion. This is the current idea and also model. The triskelion, so these different arms in there, potentially are also interacting with different interacting lipids or membrane proteins. There's a lot of research going on currently. And they also modulate this potentially as their function. Now, what has been shown already in the past 7-8 years is that calcium is one of the ions which actually is being then flowing through the channel opening. So upper mechanical stimulation, and I apologize for this rendering. Of course, it's much more sophisticated now, this transmembrane protein, but upper now mechanical stimulation, channel opening now leads then to the influx of calcium. And as you can see, there is a dramatic increase of calcium, which generates in principle a calcium nanodomain, reminiscent of the ideas which we have in neuroscience, where we talk about calcium nanodomains. So here, for example, going from approximately 100 nanomolar to more than 1 micromolar. Now, the idea for us was, how can we visualize this mechanical, the function of this channel? And this was at a time where the structures were not really much known when we started with this one. So the idea now just is to use a GCamp. So GCamp is a fluorescent protein which increases its fluorescence upon complexing calcium with calmodulin. We chose this one, or Sina chose this one, because she didn't want to have a FRET pair where it also occupies different wavelengths, which is only one wavelength, so green wavelength is being occupied. I should mention we're also doing now also red fluorescent RChem. So just to combine them potentially also with another fluorescent proteins, which are already expressed. The problem was though that the GCamps we are used to are of very high affinity because they're optimized for neuronal research. High affinity and very fast responses to somehow then be equal to electrophysiological readings, for example. Now we wanted to have something which has low affinity, given that we have a high influx of calcium. Also, second, as an a justification is to be blind to calcium fluctuations, which normally occur in the cytoplasm of the cell of interest. The second one is that we wanted to play around with the different linker links. I must say that Sina at that point also tagged them on the C or N terminus. Should mention that C and N terminus are located in the channel opening directly. So at the end we figured out this is the C terminus, but she did a lot of different modifications. So so then she started to design a Piezo1 based fluorescent reporter screen. No, what she did actually is, and this is probably something as a good advice for every PhD student, is she was fortunate enough to be invited for protein engineering workshop at Janelia Research Campus out of Washington, DC. This was due to, for us with primed conversion, I had already visiting status, a faculty position there, so she could also then join it. And there she presented her idea, and with this purpose to engage with people and to question them, the protein engineers there in the community, whether they have a GCAM with low affinity, but with a high dynamic range. And what you see on the left side in the table is she then received then 5 different versions of GCAMs. So she named them G1 to G5 with different affinities and different dynamic ranges. With these one, then she generated a variance, in this case 20. She did much, much more because she did also different linkers. But just for simplicity, we focused on this particular approach. So linker length means then attached to Piezo1, with a short, with a longer one, with some flexibility, and then cytosolic as a comparison. She applied them for this one a piezo-dependent stimuli, and this is based on shear stress, so it's a flow chamber within integrate into confocal system. And the other one is a Piezo1 independent stimuli, which is agonism, so just opening up the cell then for calcium influx. So the experimental workflow is being shown here is she transfects the cells, she just she seeds them, and then she provides them stimuli, different types of, and I was just going to mention also different chemicals that we played around for modulation the function, segments them, and then later on she performs the image analysis. So the screen for functional specificity had the desired outcome of this particular screen was to have a reporter which has a high affinity or high responsiveness to shear stress, but a very low response to ionomycin. So specific to the Piezo1 dependent stimuli, but unspecific to the Piezo independent stimuli. Now, this is part of the screen what Sina performed. And these are the intensity increases then on the y-axis, and then we have the different subpopulations. She analyzed a cytosolic, no linker, short linker, and longer linker. And then there are these different versions. And I would like only to draw your attention to G4, and then in particular this one which I circled in red. This is with one short linker. What you see then there is with the blue dots, this is the response of the sensor to shear stress, so to the piezo-dependent stimuli. And with the black dots is the response to ionomycin, so the piezo-independent stimuli. If you compare now, and there you see that it's much more responsive to the shear stress rather than to the ionomycin. As a comparison then, if you look at the eGFP, so on the last panel on the lower right, low right one, there you don't see any increase for shear stress and ionomycin. It's just GFP being attached the same way as in the G4 with the 1GSG linker. So this was very encouraging. And I should mention, if you look at the red circled one, on the right side of the red one, you see the longer link, that was 2 GSGs. And there you see that actually the specificity of the response is being lost. So actually even also then the linker length has some importance in the providing or conveying specificity. So Sina named the GenEPI. Usually I allow my PhD students, given that they are like running the projects, to name them. And she didn't come up with this one, so I had to push her. And then at some point then she came up with GenEPI. This is a linker actually in the Alps of Italy and France. So genetically encoded piezo indicator. So here you see then on the upper left side, HEC cells. So we transfected them with different cell types. And here you have HEC cells. We apply shear stress, so flow, in an imaging chamber. You have the fluorescence image on the upper left side, and the lower ones is the corresponding intensity graphs. And as you can see, there is an increase up to 1.6, 1.7 fold, and then it goes to baselines roughly after 30 seconds. So this is a systemic approach, meaning the entire cell has been subjected to a mechanical stimuli. This in the right panel, what you see then there is the increase in fluorescence with different conditions in one case. In the middle one is GenEPI again, so specific for mechanical stimuli, the shear stress and blue dots. The black dots then is down, so this is the control. And the specificity is only provided when you look at GenEPI and not for ionomycin. So what you see also there is that it does not, what we did then is, rather than doing ionomycin, we wanted also to look at physiological fluctuations of cytosolic calcium, so not so harsh conditions. So we put an ATP. And here in this case, we co-expressed and also a red calcium sensor, cytoplasmic one. So if you put then ATP on them, it increases, but it's not being recognized by GenEPI, meaning that GenEPI does not really respond to physiological fluctuations of cytosolic calcium. Oops. Yes. So what we did then is also to perform then, it has been shown by Adem Pataputian in a chemical compound screen that he identified, I think he used 3 million compounds, and identified that Yoda is a chemical agonist of GenEPI. So the application of Yoda leads to the activation of Piezo1, I should say. And as you can see, the same actually also accounts for GenEPI. So when you apply now Yoda1, there is an increase 2 to 3-fold of the fluorescence upon application of Yoda1. And this is just with fluorescence readout on the left panel. And the other panels, then there are electrophysiological recordings of wild-type piezo as well as for GenEPI. So as you can see, we applied here different concentrations of Yoda from 0.1 to 10 micromolar. You can see an increase in the response of GenEPI, now recognized with electrophysiological recording. And then you have also the EC50s, and it tells you already that GenEPI has comparable responses to wild-type Piezo1. What I showed you so far is the workflow for a systemic approach, so meaning flow shear stress, so flow, then applied to the entire cell. What we wanted to do now is another form of mechanical stimulus, so compressive force, for example. And here what we did is to transfect the cells with GenEPI, and then to seed them, and then to perform force spectroscopy, so AFM with a cantilever, just to push onto the cells. So this allows you to have, of course, very precise mechanical stimulation, a very localized area. And here are the results. So on the left side, what you see is, on the upper side is the bright field, and the lower ones you have the fluorescence. Before stimulation, after stimulation, they have the cantilever which pushes on them. You see a localized increase at the center of the cell. On the right side, you have the different forces being applied. And you can see that roughly above 200 something nanonewton. So you have an increase of GenEPI. In this case, it's 2-fold. And it reaches then the baseline, this time faster, so roughly like 10 seconds. And previously was more like 25 seconds. Now, just only what we probably should mention is, coming back on this one, probably I missed one slide here, is that it's fairly dramatic, the force which are being applied. So 250 nanonewtons, so is in a range of 50 millimeter Hg. So it is, though, in 1/3 of the physiological forces which we are being exposed in the vasculature in our body. What we then did is to do an inducible GenEPI in mouse embryonic stem cells. So in this case, it's dox-inducible. So we generated this particular line. And as you can see here, this is the outcome. On the left side, you have now the stem cells expressing GenEPI, but without induction, so without dox and without actually Yoda1. So you don't see any signal. So if you put now doxycycline, it induces the GenEPI expression. So you have this predominantly membrane label. And when you put Yoda, then you have a dramatic increase 2-3-fold. And I should mention, though, that the expression profiles of GenEPI differ between different cells. In one case, you have much, much more membrane, a little bit of ER. GenEPI is expressed in both the ER as well as also the membrane. However, when we quantify these increases, what we saw so far is when we applied our mass of quantification, either the membrane or the ER or the entire cell, there's not really much of a difference. So the next step was then to differentiate GenEPI, these iGenEPI cells, mouse embryonic stem cells, and we then used then blocking Wnt signaling pathway and then the mesodermal cell lineage. And they wanted to have then a differentiation into cardiomyocytes and to see then evaluate GenEPI's response to hemostatic cell motions. So, and this is in principle one of the results we accomplished. So what you see here is a patch of cells, so in thickness of roughly up to 300 microns, this probably being imaged like 100 microns in depth. These are cells being exposed, one cell expressing GenEPI, there are cardiomyocytes contracting, and this cell is being attached to it, and then it senses also these contractions. What you see is that it has a response time which is sub-second. So it has a higher spatial resolution and also temporal resolution. The increase only occurs actually at the extension of the cell. And it also can sense this one. So we wanted obviously whether this contraction is triggered by mechanical stimulation. And for this one, then we use then blebistatin. So blebistatin is a myosin-2 inhibitor. So this actually in principle addresses only contraction-triggered mechanical stimulation. As you can see here, then it results in the reduction of the error response, also in the frequencies, as well as also in the amplitudes, which indicates that indeed the response of GenEPI is based on mechanical stimuli. So with this one probably, yes, probably it's enough like 15, 20 minutes. What we're coming back now is to interrogate the role of mechanical stimuli asymmetry. So we have now what I mentioned to you beforehand, the lineage tracing approach with primed conversion. I should mention that we're building now, we got the grant from BBSRC with Chris Waters in our department. We're building a primed conversion light sheet, which allows us then to tag individual cells and to track them actually through development, either in mouse embryos or also in organoids. And to combine this one now with GenEPI, we are just in the process. The transgenic lines in mice are ready. We haven't tested them due to the pandemic, so we're trying now to look at them. But then really to look at individual time points during development and to see whether Piezo has a function. So Piezo is expressed. So this one is a staining of Kate McDowell. When she was still, I think, at Geneva, she shared it with Sine, these pictures. So, and it has been also implicated that YAP, for example, is downstream of Piezo. So it would be very nice if it would fit in this one, that GenEPI actually, or Piezo1 actually, the forces being sensed, either these are cell forces, cell triggered forces, cell streaming, for example, being sensed in by membrane changes, when we can image them at particular time points, and then we can track each and every cell by prime track with our prime conversion light sheet, and then potentially have a causal relationship from asymmetry early on in development and leading them also then to morula and blastocysts. We are not restricted only to this. This is one approach we would like to do. Obviously, Piezo is much more important, or potentially even physiologically, so at much later stages, so heart development, vascularization. We're also looking into zebrafish. I just got some pictures from my PhD students that we have now, some lines. This is in collaboration with Scott Holley at Yale University. So we are now eagerly testing how GenEPI actually functions also in zebrafish. The idea though is rather broad, what we are trying to approach. Obviously, one thing is what I showed you so far is more like the mechanical interrogation is what are the candidates of Piezo functions? So downstream, there has been implicated, yeah, but now we have the ability to directly look downstream. Also, what are the modulators of Piezo functions? So cytoskeleton, lipids, membrane proteins, the triskelion, how do they affect actually the responsiveness of GenEPI at different cellular context. We are now in discussions to hopefully set up a screen for Piezo-specific channel activating drugs, but also channel blocking drugs actually. So XU et al, this was, they did activating, but we are also very much keen on blocking. Ideally more the partial ones, not the massive ones, because their physiological might have a lot of side effects. And differentiation, we're just currently doing neuronal differentiations, and to look, let's say, if there are really difference between neuronal and astrocyte differentiations, and also would like to see the role in mechanics and cardiovascular development, so the role of Piezo in vivo. Well, with this one, I would like to thank you for your attention, and hopefully later on to address some questions. Thank you.